All right, howdy folks. Welcome back to Professor Carbon. In this video, we're going to go over some of the fundamental reactivity of amines, and hopefully you're familiar with the idea that the majority of nitrogens can be basic. And that, of course, is true especially for aliphatic amines or primary amines. But when you incorporate that nitrogen atom into a more complex functional group, often the basicity is decreased quite significantly. And so in this video, what I want to do is kind of talk about that distinction, and we're going to do it in the context of a number of very complicated molecules. In case you're wondering, these molecules were not chosen at random. They're actually active pharmaceutical agents, and they're some of the highest selling drugs in the US as of 2018. So what I would like you to do, along with all of the other Professor Carbon videos, is to take a moment, pause this video, and see if you can answer the question on your own by circling any nitrogen atoms that would be considered basic. And here we're going to consider basic to mean something that whose conjugate acid would have a pKa probably above 3 or so. All right, take that moment right now. Okay, welcome on back. Let's just do a quick review down here of how we consider basic nitrogens. So kind of the first base that we are probably familiar with is ammonia, which of course is the simplest nitrogen containing molecule, perhaps other than N2. In ammonia here, the nitrogen has a lone pair, and we generally would assign this the hybridization of sp3 hybridized, and that nitrogen then can be protonated perhaps into an equilibrium to make ammonia, and the pK of ammonia is around 9. So we generally consider most nitrogen atoms with this sp3 hybridized nitrogen to be basic in some form. So another great example of this is the base triethylamine. It too is sp3 hybridized, and it too has this same lone pair. All we've basically done is change up the hydrogen atoms for ethyl groups in triethylamine, and it is slightly more basic, but triethylammonium we typically might say has a pK of about 11. So pretty similar ballpark, and generally a basic amine. Now, to understand why some nitrogen atoms might not have a basic character, what we're basically going to do is try to figure out, does that nitrogen own its lone pair? So both in ammonia and in triethylamine, that nitrogen has the whole lone pair. But in a variety of other functional groups, that electron density gets spread out. And so a very classic example of that is looking at the base aniline. And aniline now still has this lone pair, but of course we can draw resonance structures where that nitrogen lone pair is delocalized into the benzene ring. And because of that, anilinium, the protonated form of aniline, has a pKa only of about 5.5. And that makes it much weaker of a base and than triethylamine would be. And the reason for that is that lone pair is delocalized. So nitrogen doesn't own the whole lone pair, and so it can't use the whole lone pair's worth of electrons to engage a proton in a basic reaction. Now, another functional group that is similar but has an even better electron withdrawing group attached to it is the amide. And in this case, now we have a carbonyl. And there's a fairly famous resonance structure of amides. That look like this. And in this amide, this is probably going to actually be the basic atom. And typically you have to go to very strong acids to protonate an amide carbonyl. So, we might say that this protonated form, which again you'll notice is now on oxygen, not nitrogen, probably has a pKa in the range of -6. And of course that can be slightly dependent on what the attached R group is, but because of that, it is a very strong acid, and that makes the amide functional group a very, very weak base. akin to perhaps chloride or bisulfate anion. There's another phenomenon with our basicity that we want to be aware of, and this is perhaps best viewed by looking at the strength of the bases of carbon. And so methyl lithium is a much stronger base than vinyl lithium, which is a much stronger base than acetylide. And the pKa's here are going to be about 25, about 43, and about 50, respectively. And what we can see here is in all three of these examples, these are carbon hydrogen containing molecules. So they're only hydrocarbons. There's no heteroatoms involved in any of them. And as you go from sp3 hybridized to sp2 hybridized to sp hybridized, the percent S character in those orbitals goes up. And it goes up from 25% to 33% to 50% over here. And this is important because, the p orbitals are kind of dumbbell shaped, right? They're long and the lobes are out here. And because of that, the electrons are further away from the nucleus. When it's S-shaped, remember that S orbital is spherical and the electrons are a little bit closer to the nucleus. And the nucleus has protons, the electrons are negatively charged. They have an affinity for positively charged protons. And because of that, as things approach the nucleus, they're less willing to engage other things out here in a basic reaction. This is important for nitrogen as well. And the nitrogen equivalents of this are to, again, look at ammonia, to look at something like an imine, or I'll put pyridine on here, and we'll talk about heterocycles in a moment. And then kind of the last one here is the nitrile. And because of this effect, nitriles are not particularly basic. They have a nucleophilicity or a basicity similar to amides. So they're in the same ballpark up here. And so as you go from that sp3 hybridized nitrogen and ammonia to something else to the nitrile, you end up really mitigating the basicity. And so these end up not being basic. All right, the last thing to discuss here is going to be heterocycles. And heterocycles are quite annoying for a variety of reasons, and it's going to depend a lot on what the how that heterocycle is set up, if we consider it a basic nitrogen or not. So in pyridine here, the conjugate acid of pyridine is pyridinium. It has a pKa of about 5.5. And because of that, we consider pyridine to be a base, or basic N. And if you go through and look at the pyridine molecule, you'll notice that this has a very key imine. So there is a C double bond N in there. And what that's telling us is if we look at the structure of pyridine, there already is a pi bond here between the nitrogen and the carbon. And that means that, in the sp2 framework, we have the sigma bonds or single bonds, and then we have the pi system or the pi cloud. And the pi cloud is always going to be opposed to or orthogonal to that sigma framework. And in this case, that N lone pair is part of the sigma framework. So it's actually in an sp2 orbital. It's not part of a pi system or pi orbital. And because of that, it's pointing out of the ring, and it is available to engage in H+ in an acid-base reaction. This is going to contrast quite markedly with the 5-membered heterocycle pyrrole. And you'll notice in this case, we draw an N lone pair on nitrogen. It does not have a C double bond N. And if we look at the structure of that pyrrole on the side, each of these carbon atoms have their p orbital for a pi bond. But to get to the aromatic number of 6 pi electrons, we need to engage this nitrogen lone pair. And so this lone pair is in the pi cloud. And because of that, it's delocalized. And if it's delocalized, it is not going to be, it's not going to be basic. And this is probably the most confusing of these to identify. But what I encourage people to do is to look for that C double bond N. So most of the things that have an explicitly drawn C double bond N, we will consider basic. And most of the things that have 3 separate bonds to nitrogen and are part of a heterocycle, we'll consider non-basic as long as they have an aromatic number of electrons. So with this review, let's go back and check out our molecules up above and see if we can identify where all these nitrogens are. And I'm going to use red for not basic, and then we'll use green for basic. Okay, so looking at the nitrogen here, when I make this first glance, I see that it's part of an amide, and I would say not basic, because it's part of that double bond character. And if I ask, where are my electrons on that nitrogen, they're going to be going up into the carbonyl. And even worse, we could draw some resonance structures into this adjacent aromatic ring system. And so this thing is going to be even less basic than your regular amide because some of those nitrogen electrons have 2 places to go. Same is going to be true on the other side here. So we got this nitrogen. It too is part of this amide carbonyl system, and it too has resonance delocalization into this benzene ring. And so neither of these I would consider to be basic nitrogens. Same is going to be true here. And let's just go through and maybe sketch out all of our amides in all of these structures. So here's another amide. So that one's not going to be basic. Here's another amide, so that one's not going to be basic. This one, again, you'll notice it has the same problems. It has plenty of places for that N lone pair to go, both into the carbonyl over here, into the benzene ring on this side. And believe it or not, it can go into this S double bond C over on the far side. Same is going to be true for this nitrogen. It can go into the S double bond C. It can go into the benzene ring, etc. Again, we have an amide on this side. In this case, though, it'll be slightly better because we don't have a benzene ring to delocalize that N lone pair into. On the last molecule here, again, we have an amide, so we're going to exclude the amide basicity. All right, so that at least gives us a lot of things to think about. Let's just do a quick dive through with the green one and circle anything that obviously is a strong base. Nope, looking here, so this one is definitely going to be a strong base because it is a primary amine, and it's a primary amine with an sp3 carbon next door, which means there's no place for that N lone pair to go. So this nitrogen, of all of the nitrogens drawn on these 4 molecules, is probably going to be one of, if not the most basic. The other one that is a really good contender is this one, because it too is an sp3 nitrogen, and it too is flanked by good old saturated carbon on both sides, sp3 hybridized carbon. And so that nitrogen fully owns its own lone pair, and because it's a secondary amine instead of a primary amine, it probably actually will be more basic than the one down below, but by a very small amount, okay? A single pK unit or maybe less. Okay, thinking about all of these other nitrogens now we got a whole lot to consider. Let's exclude one here right off the bat. That's easy. So remember, this is our C triple bond N that is sp hybridized. It has a very high % S character, so it will not be basic. What's surprising then is none on here are basic. So that's pretty remarkable, I think. This molecule's got 1, 2, 3, 4 nitrogen atoms, and I don't think I would consider any of them to be a strong base. All right, so let's see here now. Up top, let's review our 5-membered heterocycle. So any 5-membered heterocycle here with the nitrogen having 3 bonds will not be basic. So, that would be this one here. is again that same motif. Now, of course, there's a bonus nitrogen in there, but that doesn't matter. It's, you know, really trying to get up to that 6 electrons, and we've got 2 electrons from this double bond, 2 electrons from this double bond, and that means to get to the 6 electrons, we got to use the lone pair on nitrogen. Okay, so that one is not going to be basic. Let's see here, looking over, there's none on there that we can exclude. And down here, we have one that we can exclude in a similar way. So again, we've got 2 electrons, 4 electrons from that double bond. We need 6 electrons here. So this nitrogen is not going to be basic either. All right, so that means we're done with this one. We're done with this one. Oh, nope, we got two up top. All right, thinking through the rest here, let's dive into this one. Okay, so now this one, if we look at it, we're going to have this double bond N, and we don't really care what the rest of it is, because this nitrogen, because it already has that double bond to carbon, that means that the C double bond N has got to be part of the pi system. And this means that this has to be part of an sp2 orbital sticking out of the pi system. And so any of these with these C double bond ends have to be basic. So that one's good there. Now we can really start assigning these. That one's going to be basic. This one's going to be basic. This one's going to be basic. Now we're really rocking and rolling. And that helps us here too. So now we've got, that one is going to be basic, and this one's going to be basic. Now for the advanced students, I'll point out that this CF3 is next door is really going to diminish the basicity of these amine nitrogens, but for now we'll pretend that they indeed could be some kind of a base, because that lone pair is going to be sticking out from the pi system. Okay, now we're done down here, so that's good. We're done down here, so that's good. We're done over here, so that's good. Now we have 2 pesky nitrogens to consider over yonder, these 2 here. And I left these 2 for last on purpose. This one on the bottom here, I'm going to say is probably going to be basic. So it, to me, looks like an aniline, but unlike this one, we're not going to be able to get that nitrogen lone pair to fully delocalize into the nitrogen. So I will circle that one and call it basic. It's going to be a weaker base, just like how I indicated aniline has a lower pKa than triethylamine, but it'll still probably qualify. Certainly it'll be less basic than the other one on the other side of the piperazine. Okay, so what I wanted to point out about this last nitrogen here circled in yellow, kind of smack dab in the middle of the molecule, is in principle, this one should be basic. It looks aniline-like, it's not an amide. In reality, though, it's going to be really difficult to get that one to protonate. And the reason is if we consider molecules like 2-aminopyridine, and if this molecule protonates, we can make the pyridinium, and then you can have a resonance structure. And because of this, the nitrogen at the 2 position of these pyridine-like motifs, and that's what we've got here. If I erase this all, clean up my mess a little bit. So this nitrogen in the middle is not really going to be owning its lone pair all the way. It's going to be delocalizing it into not 1, but 3 different nitrogens that are all, quote, or ortho-like on these adjacent heterocycles. And so because of that, these heterocycles will be more basic than they might otherwise be, but that nitrogen in the middle won't be. And so that's kind of an advanced twist that's special to this particular 2-amino or 4-amino, I'll point out. And there's a very famous base that takes advantage of this called dimethylaminopyridine. And we usually call it DMAP for short. And in DMAP, this is the important basic component. And the pK of DMAP, protonated DMAP, DMAPium, if you will, is going to actually be closer to 9. So it gets almost 4 pKa unit bounces by having this dimethylamine in the backbone. So with that, I'd like to thank you for tuning into this video. As a reminder, everything in the playlists is sorted and color-coded. So you can look for the kind of functional group or chapter you're looking for up here, and you can look for the kind of problem you're trying to study over here on the screen. So hopefully you're getting use out of the Professor Carbon videos. Please give us a thumbs up or like and subscribe to the channel.